Part 1. Please find a quiet place to sit or to lie and to listen to this audio. Once seated or lying, close your eyes. Focus your attention on your breath. When you're ready, inhale slowly. Let the air fill your lungs. And then exhale slowly. Let the air escape from your lungs. Inhale again, and then exhale again. Notice how your stomach rises as you inhale and falls as you exhale. Rises as you inhale and falls as you exhale. If your mind wanders, don't be concerned. Just gradually shift your attention back to your breath. You might notice how many thoughts enter your mind, randomly and haphazardly, like a throng of emails. Perhaps you can describe your thoughts. For example, you might be experiencing the thought that, I hope this audio is not a waste of time. Describe this thought. That is, repeat to yourself something like, I'm experiencing the thought that I hope this audio is not the waste of time. Or you might be experiencing the thought that my neck is sore. Describe this thought as well. That is, repeat to yourself, I'm experiencing the thought that my neck is sore. Now continue this exercise for a while. That is, repeat to yourself, I'm experiencing the thought that, and then describe your thought. Now, as you complete this exercise, you might realise that thoughts don't always reflect reality. They're merely thoughts, fleeting and intangible events. The thought that perhaps you're not intelligent does not indicate that you are not intelligent. The thought that perhaps you're not worthwhile does not indicate that you're not worthwhile. These thoughts are merely fleeting and intangible events. Part 3. Now generate an image of a brain operating inside someone's head. Notice all the cracks and crevices and ridges that cover this brain. Don't worry if your image is hazy or unstable. If the image vanishes, just form the image again as best as you can. Now imagine you can see the cells in this brain in more detail. Imagine the brain is composed of billions of cells. Many of these cells, called neurons, resemble tiny strands of spaghetti. But now focus your attention on one of these strands, one of these neurons, as though peering through a powerful microscope. You notice that each neuron is more complex. Midway you notice a bulge or sphere called the cell body. Sprouting from one end of this sphere or cell body is a network of thin branches like a tree called dendrites. And sprouting from the other end of this sphere or cell body is a long strand like a hose called an axon. Maintain this image of the neuron for a minute. Repeat the word cell body as you shift your attention to the bulge. Cell body, cell body. And repeat the word dendrites as you shift your attention to the network of branches. Dendrites, dendrites. And repeat the word axon as you shift your attention to the long hose. Axon, axon. You don't need to memorize these words now. Indeed, you don't need to devote any effort into this exercise. You merely need to follow the instructions as best you can. Part 4. Now shift your attention to the long axon or hose that emanates from the sphere or cell body. Toward the ends of this hose or axon, that is at the opposite end from the cell body, are several branches. Maybe you can see 3 or 4 branches, maybe you can see 9 or 10 branches. Regardless, focus your attention on one of these branches. You notice that each branch seems to end near another neuron. More precisely, each branch seems to end near the dendrite of another neuron. Perhaps one branch ends near the dendrite of one neuron, and perhaps another branch ends at the dendrite of another neuron, or perhaps another dendrite of the same neuron. Part 5. Now zoom your attention towards the end of one branch of this axon. at the point at which this axon seems to enjoin a dendrite of the next neuron, as if peering through a very powerful microscope. You may notice both the axon and dendrite look slightly different to what you expected. The end of this axon seems to bulge slightly, called the axon terminal, and the dendrite seems to contain lots of little spines, like the sharp nettles on a pink rose bush. Now, the end of this axon is located very close to one of these spines. This juncture between this axon terminal and the dendrite of another neuron is called a synapse. As you maintain this image of sort of the bulging axon next to a dendritic spine, repeat the word synapse a few times. Synapse, synapse, synapse. Notice how the axon does not quite touch this spine. Instead, the axon seems to end just before the spine. A small gap separates the axon and the spine, and this gap or juncture is called the synaptic cleft. Don't worry if your image is not quite accurate. Just maintain an image of a cleft or gap between the end of one axon and the dendrite of another neuron. Part 6. You then realise that both the terminal of this axon and the cleft or gap between this axon and the next neuron is abuzz with activity. Some molecules or particles like glowing M&Ms seem to emanate from the axon terminal. Other molecules seem to swim rapidly around this gap, and a few molecules seem to attach to the dendritic spine of the next neuron. So what is happening, you wonder? What is the role or purpose of these molecules or particles? First, you notice that inside the axon, especially near the end or terminal, many chemicals or molecules seem to be changing. Perhaps you could imagine yellow and blue molecules like M&Ms sticking together to generate a stack resembling a Jenga construction. Or you might imagine a stack of green and red molecules dividing into smaller pieces. You notice that inside the axon, the cell is creating many of these chemicals or molecules. Soon you will learn the role of these molecules, and these molecules are called neurotransmitters. Neurotransmitters. But for now, just imagine that many new molecules are forming in the axon, especially near the terminal. And these molecules, resembling blends of M&Ms, are usually stored in vessels like tubs. And these tubs, however, tend to be spherical, like a ball called vesicles. Part 7. You can probably imagine these molecules resting peacefully and languidly in these spherical vesicles close to the end of this axon. And then suddenly an event transpires. A spark of energy, called an action potential, seems to surge down the axon from near the cell body to the end or terminal. Perhaps you might imagine a flash of bright yellow light rocket down the axon towards the terminal. As soon as this flash of bright yellow light reaches the end of this axon, a sequence of events transpires. In essence, small channels snap open. These channels resemble small toilet rolls, connecting the inside of this cell to the outside of this cell. Usually these channels are closed, as if the toilet roll has been scrunched. But after these sparks of energy reach the end of this axon, the toilet rolls open abruptly. Small particles called calcium ions then bolt into the cell through the channel. You can visualize these calcium ions as like marbles dropping through the toilet roll into the axon terminal. And as soon as these calcium ions or marbles enter the cell, the vesicles that store the molecules or neurotransmitters sort of burst open, and these molecules are then released into the synaptic cleft, the gap that separates the end of this axon from the dendrite of another neuron. Perhaps you can imagine this molecule or neurotransmitter resembling a tiny stack of M&Ms fixed together, swimming vigorously from the axon terminal into the cleft and then towards the dendrite of another neuron. And perhaps you can even hear the sound of splashing as the molecule strives to reach this dendrite of another neuron. And eventually this molecule or neurotransmitter reaches this dendrite. Often this molecule or neurotransmitter will then attach to a special location on this dendrite, a location called a receptor. Perhaps visualize the receptor as a tiny set of uneven columns, like the grand entrance of a lavish hotel after a savage earthquake. The columns are not distributed evenly. One or two columns might be upright, some other columns might be leaning to the left or leaning to the right. The molecule or neurotransmitter, if the right shape, can then sort of wedge itself within this set of columns. And when this molecule, resembling a tiny stack of M&Ms fixed together, attaches to this set of columns, or this receptor, another sequence of changes unfolds. So what is this sequence of events? What happens when the molecule or neurotransmitter attaches to the receptor? Well, sometimes nothing transpires. And sometimes this molecule will ignite a spark of energy and action potential in this second neuron. In other words, a spark of energy that surges down one axon will ultimately cause a spark of energy to surge down a second neuron and axon. Part 8. How do the neurotransmitters, after attaching to receptors, initiate this spark of energy? Scientists have discovered two distinct sequences of events. In one sequence, once the neurotransmitter or stack of NMNs attaches to the receptor or series of columns, these columns then shift. Perhaps you can imagine a creaking sound as these columns rotate gradually like an old rusty door. Then as these columns rotate, a channel seems to open, and then sometimes sodium atoms bolt into this channel. And these sodium ions then initiate a spark of energy or an action potential down the axon. Now, if you understand action potentials, you might understand how these sodium ions change the voltage of this cell, generating an action potential. But don't worry if you don't understand this argument. Don't be concerned. Just accept that neurotransmitters, after they clasp onto a receptor, can open these channels and initiate a spark of energy. And this sequence of events is called an ionotropic effect. An ionotropic effect. An ionotropic effect. Now, in a second sequence, once the neurotransmitters or stacks of M&Ms attach to the receptor or series of columns, these columns again shift. But in this case, as the columns shift or rotate, no channel opens. Instead, you can imagine something like a coil of ribbon hanging on one of these columns inside this cell collapse to the floor. And this coil of ribbon is called a G protein. And this coil of ribbon then attracts other molecules called secondary messengers. And perhaps you can imagine a raft of like tiny little servants all arriving to retrieve this coil of ribbon, or G protein, that fell to the floor. And these secondary messages then scamper down the dendrite towards the cell body, the bulge in the cell. As they approach the cell body, they elicit many gradual changes. They might open some other channels or cause other modifications to the cell, many of which ultimately might culminate in sparks of energy or action potentials. Again, don't worry if this sequence of events is unclear. Just accept that neurotransmitters, after they clasp onto a receptor, might sometimes compel other molecules called secondary messengers to shift to other areas of the cell. And this sequence of events is called a metabotropic effect. A metabotropic effect. A metabotropic effect. In short, these images may later help you understand the sequence of events at synapses. You might now understand how action potentials at one neuron produce neurotransmitters or molecules that attach to receptors and sometimes elicit action potentials in other neurons. Or you might not understand this sequence of events, but don't worry. These images will help you appreciate the material later.